Hello. So my voice is audible. So I am going to start today's topic, which is on nucleic acids. Okay, good. So it's audible. Okay. Just waiting for a few more students to join, then we will start the session. In the meanwhile, have you any question or query? Then please let me know. I'm sorry for the trouble. I don't know why the connection gets immediately break. I don't know, but right now it's okay. I think so. So please let me know if you have any question regarding the review paper. You can ask me right now. If you have any query, please let me know. If you have any query regarding the review paper, please let me know. Otherwise, by 10th of December, you have to send me your abstracts along with the title. So if you have any query, please let me know. Yes, Radha, please. You can select the case study of any disease. Please pursue it. Please proceed it. And China, yes, you can go with Leninger. Leninger is a good book. You can go with the new edition of Leninger. So please go with Leninger for this biochemistry lecture series. No, this is not MBBT. Rather, this is a biochemistry lecture series. So you can go along with the Leninger book. So shall we start now? You have any another question? Pooja, Diksha, Shaina, any of you? If you have any question, please let me know right now, because I am waiting the students so that they will also join with us. I am just waiting for them. It is 3:15 right now. Otherwise, I will start this class. Yeah, this is my email ID. You can send me here everything, and I will definitely reply. You have to think fastly. You have to do it fastly. You have to select the topic as soon as possible. Okay. So shall we start now? We have already 15 minutes right now. We get online by 15 minutes. Shall we start now? I think they will join if they want to. I will put it in the group again. Okay. So today we have to start our first lecture on nucleic acids. It's a very simple, you can say very basic of biochemistry. So we have to start with nucleic acids, structure and properties of purines, pyrimidines, nucleosides and nucleotides. Then we have structure of DNA, RNA, central dogma. Then we have to do protein hormones, steroid hormones, thyroid functions, enzymes, enzyme kinetics, enzyme regulation, lipids, fatty acids, and lots of metabolism which are there, protein structure. But on weekends, like on Saturdays and Sundays, I will try to cover two topics together, like enzymes. So enzymes, these two are together. So I will try to do it together. Like enzyme session, I will try to cover it together in one day for 2 hours. So we will see how it will work. So this PPT is visible to all of you. Please let me know. Is it visible? All of you please reply when I ask any question, anything you have to reply. Yes. Tejaswini, yes. Welcome Tejaswini. How are you? So all of you have to reply, dear. All of you, please. Yes. Good. We have Sonal, we have Afreen, we have Siddhartha, Anjali, Unnati, Radha, okay. Tejaswini. Okay, so now I will start, and by one or two slides I will came back here on this slide platform, and I will check if you have any problem, then we will discuss. Okay, so I'm just now going to start it. Yeah. So nucleic acids, what are basically nucleic acids? What are they basically? They are the long chain polymers of nucleotides which are present in a nuclei of a cell. So nucleic acid, which is basically the DNA or the RNA, nucleic acids are long chain polymers of nucleotides present in the nuclei of a cell. So suppose this is the cell. In the cell we have a nucleus. So inside the nucleus right here we have the genetic material, which is DNA and RNA. So they are also called polynucleotides. They play an important role in transmission of hereditary characteristics and biosynthesis of proteins. So what basically DNA and RNA do, or you can say the nucleic acids do, they help in the transmission of hereditary characters and in the biosynthesis of proteins. These are the largest and most complex organic molecules. So this is the history from when we are able to come about nucleic acids. Like in 1866, a scientist, which is Mendel, who published his experiments in plant hybridization, which lays out the basic theory of genetics. But it is up to, right from 1866 up to 2001, we are able to get that there is something which is known as DNA, which is known as RNA, which contains the genetic material, which contains the hereditary information. Even in 1953, we had a scientist whose name is Watson and Crick. So Watson and Crick determined the structure of DNA molecule, which leads directly to knowledge of how it replicates. So it determined the structure of DNA molecule, which leads directly to the knowledge of how it replicates. Again in 1966 we are able to know about genetic code. So we will discuss genetic code in detail also. So from 1866 we are able to get about, or we are able to know about nucleic acids. So in nucleic acids, or you can say nucleic acid basically consists of DNA and RNA, but there is one more term which is known as nucleotides. Now what is nucleotides? Nucleotides are the organic compounds made up of phosphate group, nitrogenous base and a sugar molecule. So we have, let me show you. So we have a phosphate group, which is not shown here. These are only sugars. So we have a phosphate group, we have the nitrogenous bases, which are basically purines and pyrimidines, and a sugar molecule. So these are the building blocks of nucleic acids, which is basically the DNA and RNA. They serve as source of chemical energy, participates in cellular signaling, and function as important cofactors of enzymatic reactions. Getting my point? So in history, so in 1869, we have the scientist. सर, अरे वो तो मेरी बात हो गई थी इन वाले बच्चों से, ये तो सारे आए. अरे कल तो बात हुई सारे. चलो अभी मैं ले रही हूँ, उसके बाद मैं बात करती हूँ. कोई बात नहीं, फिर मैं नहीं करूँगी. ठीक है ठीक है ठीक है ठीक है ठीक, मैं समझ गई. ठीक है ठीक है ठीक है. ओके. So the history it consists of, in 1869 we have the scientist whose name is Mr. who discovered nuclein or DNA in the cells from the pus and later he separated it into a protein and an acid molecule. So in 1869, by 18th century, we are able to know about nucleic acid, which is basically DNA and RNA, but we are able to know about it in 1869. And in 1926, there is a one scientist whose name is Levine, who proposed tetranucleotide theory, which states that nucleic acid consists of only 4 nitrides as it gives 4 different nucleotides on hydrolysis. Then again in 1950 we have another scientist whose name is Chargaff, who also proposed one theory according to which the 4 nucleotides are not present in the nucleic acid in stable properties. In 1957, we have another scientist whose name is Todd of Cambridge University, which gives the structure of nucleotide. So there is one term which is known as nucleotide. Basically, nucleotide consists of a sugar molecule, nitrogenous bases, as well as phosphate group. So up to now, is it clear to all of you? Please let me know. Is it clear to all of you? Are you able to understand it or not? My volume is okay, everything is fine till now. So nucleotides, it generally consists of a sugar molecule, a phosphate group and nitrogenous bases. So I am just doing very basic, it is a very basic lecture basically, but we have to start from it now. So okay. So just I am now, I hope all of you will be there right now because we are just starts right now. So I hope all of you don't have any problem regarding the timing. The timing is okay. The timing is okay right now. Are you able to get it or not, or the timings are okay for all of you or not? Please let me know the timings are okay for all of you because some of you are saying we will start just by 3:15 because right before we had some network issues also. Okay. The timing is okay. We are just doing it 15 or you can say 15 to just 20 minutes before. So I hope you all will understand. Okay. Okay, good. If any of you have any problem regarding the timing, please let me know. If any one of you has any problem regarding the timing, you can tell me at any time. No issues, no problem. But you have to share this thing along with me, okay? But I think this timing is okay. Otherwise, we will shift the timings also. No issues at all. Okay. So just I just let you know about the nucleotides. So the nucleotides consist of, it basically consists of sugar molecules. Like we have two types of sugar molecules. We have ribose sugar and we have 2-deoxyribose. So they are just present in fernose form, which is the form of sugar. So we have two main types of sugar. One we have ribose sugar and another we have 2-deoxyribose. So we have two types of sugar molecules and the 2-deoxyribose generally lacks the OH group. If you are able to see here, everything is same in both the sugar molecules, only in the absence of this OH group here. So here the OH group gets removed and here it is only H. So that is why it is 2-deoxy. That means it lacks the OH group. So it lacks the OH group. And now we have bases. So we have sugar molecules. Along with sugar molecule, we are just discussing about nucleotides. So nucleotides are the organic compounds which consist of phosphate group, nitrogenous bases and sugar molecule. So we just discussed about the sugar molecule, that we have ribose sugar as well as we have 2-deoxyribose sugar. Then the bases which we have two types of bases which are known as purines and pyrimidines. Now this is your homework. All of you have to let me know what is the difference between purines and pyrimidines. Like we have adenine and guanine which are purines, and we have cytosine, uracil and thiamine which are pyrimidines. So we have two types of nitrogenous bases. We have purines as well as we have pyrimidines. So adenine and guanine, they are the two purines, and cytosine, uracil and thiamine. Cytosine, uracil and thiamine, they are the pyrimidines. So we have two purines which is adenine and guanine. So you have to answer me, this is your homework, what is the difference between purines and pyrimidines? Okay. So yes, the purines which is adenine and guanine occur in both DNA and RNA, that means in both nucleic acids. But in case of pyrimidines, we have cytosine, uracil and thiamine. In case of DNA, thiamine is there. And in case of RNA, uracil is present instead of thiamine. So if you are able to see here, in the DNA, we have adenine, guanine, cytosine and thiamine. In the RNA, we have adenine, guanine, cytosine and uracil. So our thiamine, which is our pyrimidine, it gets replaced by another pyrimidine which is uracil. Getting my point? So we have two types of sugar molecules. We have D-ribose and we have 2-deoxyribose. Number one. Then we have two types of bases, nitrogenous bases. We have purines as well as we have pyrimidines. So purines consist of adenine and guanine, and pyrimidine consists of cytosine, uracil and thiamine. So this is the simple, you can say, purines and pyrimidines. This is adenine. It consists of a double ring structure, and in case of cytosine, which is the pyrimidine, it is a single ring structure. Getting my point? So this is the difference between purines and pyrimidines, that they are double ring structure and it is a single ring structure only. So we have adenine, guanine and we have cytosine, uracil or thiamine as a pyrimidine. So these are the things. We have 5-methyl cytidine, we have inosine. So these are another, you can say, nitrogenous bases which are present. We have 5-methyl cytidine occur in the DNA of animals and higher plants, and 6-methyl adenosine occurs in bacterial DNA. So this is the nucleic acid which we have to study. It generally consists of sugar molecule, phosphate group as well as nitrogenous bases. So the nitrogenous bases, we have purines and pyrimidines. In sugar molecule, either it is ribose sugar or deoxyribose. Along with it, we have a phosphate group which gets attached to it. So all these things together make the nucleosides. So we have three terms: nucleosides, nucleotides and nucleic acids. So any one of you, please let me know the difference between nucleosides and nucleotides. Any one of you, please. Any one of you, please let me know about the difference between purines and pyrimidines, as well as the difference between nucleosides, nucleotides and nucleic acids. Any one of you, please. Please tell me. Yes, yes, Kalpesh, very good, very good. Please, any one of you, let me know. Yeah, very good, very good. So, nucleoside is basically a nucleotide without the phosphate group. And what is the difference between purines and pyrimidines? Any one of you? What is the difference between purines and pyrimidines? Nucleotide is only saying nucleotide is sugar plus nitrogenous base plus phosphate group. Yes, good, good. China is saying double ring purines. Yeah. So we have purines which is double ring structure, and we have pyrimidine which is single ring structure. Ha. So nucleotides have three things: sugar, phosphate group along with the nitrogenous bases. But in case of nucleosides, we have only sugar and nucleoside bases, but absence of phosphate group. Yeah. Sonal is saying nucleic acid is a long chain of nucleotides. Very good, very good. Radha is saying nucleotide is sugar plus phosphate plus nitrogenous bases. Yes. So what is the difference between pyrimidines and purines, Radha? Purines have double ring structure, and pyrimidines, and any another difference irrespective of the double ring and single ring, any another difference? Yes. Yes, Marilyn is saying purine occur in DNA and RNA. Very good. In case of pyrimidines, either we have uracil or we have thiamine. Ah, yes. Diksha is saying purine has high melting point and boiling point. Now the question arises, why the purines have high melting and boiling point? Why? The purines are double ring structure. They have high melting and boiling point, but why? Why they have high melting and boiling point? Any one of you, please. Definitely Sonal. Sonal is saying they are high molecular weight. Okay, any another thing? China. China Gautam saying double ring gives them high resonance which makes them stable and hard to break. Okay, very good China, very good. Double ring, yes, because they have a double ring structure, double ring, that is why they have high melting and boiling point, or you can say they are structurally little bit more complicated. Yes, yes, all of you are very right. Good, good. Ayushi, dear, we will discuss about this later on. Just focus on the theory class right now. I will give you 5 to 10 minutes after the lecture, then we will discuss about it. Okay? No issues, please, don't worry. So, we have nucleosides, nucleotides, and nucleic acids. So nucleic acids are the third class of biopolymers, which are basically polysaccharides and proteins. Then we have two major types of nucleic acids. We have either DNA or we have RNA. Now what is DNA? Basically you all know DNA is deoxyribonucleic acid, which is the carrier of genetic information. Similarly, RNA is ribonucleic acid, which generally helps in passing the information from DNA, or taking the information from, you can say, taking the information from the DNA, and helps in making the proteins also. So ribonucleic acid or RNA, an intermediate in the expression of genetic information and other diverse roles. We have one more term which is central dogma. So what is central dogma? So when we have DNA, from DNA we are able to get mRNA, which is messenger RNA, with the help of process which is known as transcription, and from mRNA we are able to get proteins with the help of translation. The whole of this process from DNA, when we are able to get the proteins, this is known as central dogma. Getting my point? So the monomeric units for nucleic acids are nucleotides. Nucleotides are made up of three structural units, subunits. We have sugar, we have nitrogenous bases, and we have phosphodiester, or you can say phosphate group along with it. So nucleoside is only sugar plus base, the nitrogenous bases. Nucleotide is sugar, nitrogenous bases along with the phosphate group which is attached. So we have nucleosides, we have nucleotides. So nucleotide consists of sugar along with base and the phosphate group attached. Now, what is nucleic acid? So, nucleic acid is the polynucleotides, so that they will join together. So, we have sugar, base and phosphodiester. One nucleotide, it attaches with the another nucleotide and they form the long chain together. So, they form the long chain together, then it becomes the nucleic acids. So, again we have pyrimidines and purines. So we have adenine and guanine as a purine and cytosine, thymine and uracil as pyrimidine. So now we will discuss little bit about the sugar which we have. We have two types of sugar, simple ribose sugar and we have 2-deoxyribose sugar. So what are they basically? The sugar is nucleotide and so in nucleic acid is a pentose. So we have 5 carbon sugar. In RNA and its nucleotide, the sugar is ribose, whereas in DNA and its nucleotide, it is deoxyribose. So, it is without the OH group. So, right here we have the OH group which is present in case of ribose, but in the case of 2-deoxyribose, OH group is absent. So, in RNA and its nucleotide, the sugar is ribose, whereas in DNA and its nucleotide, it's deoxyribose, or you can say without oxygen. So, nucleotides can be hydrolyzed to yield nucleosides and phosphoric acid. So, we have, in case of ribose, we have the presence of OH, and in case of 2-deoxyribose, there is a lack of OH group. So this is the structure, how the nucleic acids, how they will bond with each other. So this is the sugar molecule, the ribose sugar, along with phosphate group on one side and nitrogenous bases on the another side. So this is adenine, like their cytosine, thymine. So we have purines, pyrimidines. They will, you can say, they will form the hydrogen bonds and interact with each other. So in adenine and thymine, we have a double bond. In case of cytosine and guanine, we have the triple bond. So adenine and thymine, we have a double bond. So the parts of a DNA molecule, two polynucleotides. So two polynucleotides or two chains running in opposite directions are held together by hydrogen bond between the bases. So we have the hydrogen bonding between the bases. A links with T, that means adenine always bind with the thymine. So one purine will bind with the thymine, one will bind with adenine will bind with thymine by two hydrogen bonds, and cytosine will bind with guanine with 3 hydrogen bonds. This is complementary base pairing. So this is the complementary base pairing. So if you are able to see here again the structure, as we all know, DNA is double helical structure, RNA is single helical. So when two polynucleotide chains are joined together, they will form the base pair. They will form the bonds between the nitrogenous bases. So adenine will bind with thymine, cytosine will bind with guanine. If you are able to see here also, we have sugar molecule, and on the one side we have the nitrogenous base, and on the another side we have the phosphate group. So we have the sugar molecule. One side we have the nitrogenous bases, which is adenine, cytosine, guanine, thymine, and on the other side we have the phosphate group which is attached. So this is the pentose sugar. So this is basically the double helical structure on which we are working right now. So what is DNA basically? DNA is deoxyribonucleotide. It is the monomeric building block of DNA. A phosphate group and a nitrogenous base both bonded to a deoxyribose sugar. So that is why it is known as deoxyribose, because it lacks the OH group. Getting my point? And in case of ribonucleotide, monomeric building block of RNA, a phosphate group and a nitrogenous base both bounded to the ribose sugar. That is why it is known as ribonucleotide, because they have the ribose sugar which is present. So duplex is two complementary strands of DNA. Mutant is genetically altered species or cell. We already discussed nucleosides, nucleotides, nucleic acids, and yes, phosphodiester bond. Then there is one more term which is known as replication. Now what is replication? Any one of you? Please let me know what is replication. What is the difference between replication, translation and transcription? Any one of you? Any one of you? What is the difference between uh, replication, transcription and translation? What is replication basically? Replication, any one of you please? Yes, yes, Shaina. So replication is a replica. When we are able to get the similar copy of the original one, it is known as replication. So replication is DNA to DNA, or you can say you can get the copy of the original one. And what is transcription and translation? All of you, please, all of you reply. What is the difference between transcription and translation? Yes, yes. Radha is saying replication means copying. Anjali is saying transcription or DNA to RNA. Paushali is saying DNA to mRNA is transcription. mRNA to protein is translation. Yes. Shraddha is very right. Transcription is from DNA to messenger RNA. And translation is from messenger RNA to tRNA, transfer RNA. So these are very basic terms which are there: replication, transcription and translation. Replication is the DNA synthesis, or you can say copying of the original sequence. Transcription is synthesis of mRNA from DNA, and translation is synthesis of proteins from the RNA template. Now what is template basically? What is template? What is template? So a template is a section of a DNA which is being replicated. Suppose this is the, you can say this is the double helical structure. Suppose. So when we are working on this structure, when we are able to get the messenger RNA, when we are able to get the RNA, and then we will get the proteins. So when we are working on a particular DNA sequence or a segment, it is known as the template. So a section of DNA which is being replicated or transcribed, mRNA which is being translated. So template is basically the particular section or a particular segment on which we are working on. This is again the nucleosides, which is basically consists of sugar and the nitrogenous bases. Now the phosphate group is generally negatively charged. You all know that phosphate group is negatively charged at neutral pH, typically attached to the 5' position. Nucleic acids contain one phosphate per nucleotide. So this is basically the whole nucleotide. This is basically the base of the biochemistry, the base of the nucleic acid, the DNA, the RNA. It generally consists of the pentose sugar along with the phosphate and along with the nitrogenous bases. So this is the carbon 5 carbon ribose sugar, which is basically deoxyribose. So we have 5 carbon sugar along with nitrogenous bases on one side and along with phosphate group present on the another side. So tomorrow I will check whether you have draw all these nitrogenous bases. You have to draw all the structures of these nitrogenous bases, the structure of nucleotides, the structure of nucleosides, and of course this polynucleotide also. So you have to draw all these structures. Okay? You have to draw all these structures of nucleotides, nucleosides, nucleic acid, everything you have to draw. So similarly, this is the DNA. If you are able to see on this side, we have the RNA. So DNA consists of ribose sugar, deoxyribose. If you are able to see, there is the absence of OH group. So we have ribose sugar, deoxyribose sugar along with the phosphate group and nitrogenous bases on the another hydrogen, or you can say on the another carbon. So we have guanine, thymine, adenine. Similarly in RNA, instead of thymine we have uracil. So you have to draw all these structures. This is again nucleotide monomers are joined by 3' 5' phosphodiester linkages to form nucleic acids, or you can say polynucleotide polymers. So the ribose sugar along with uracil, guanine, cytosine, adenine on one side, and we have the phosphate groups on the another side. So you have to draw the structures of DNA as well as the RNA. And yes, one more important thing is that we have the bonding between two polynucleotides, either double bonding or we have the triple bonding. So when adenine always pairs with thymine, so when making hydrogen bond, cytosine always pair with guanine. So you have to mind this thing that adenine pair with thymine, cytosine will pair with the guanine. So adenine always pair with thymine with double bond. Cytosine always pair with guanine with triple bond. So you have to remember this thing. That adenine will pair with thymine, or you can say with uracil also. So adenine will pair with thymine, cytosine will pair with guanine. Yes. These are again the same thing. We have adenine, guanine, thymine, cytosine and uracil. Cytosine and uracil in RNA. Cytosine and thymine in case of DNA. And yes, we have one more linkage which is there, which is known as beta glycosidic linkage, which is between the sugar molecule and between the nitrogenous bases. So we have beta glycosidic linkage. So you have to draw all the structures of these purines and pyrimidines. Like pyrimidines are single six-sided ring structures, and purines are double ring structures. These are some of the properties of the nitrogenous bases like purines and pyrimidines. Some of you have said that they have high melting point and boiling point. So they have some properties of purine bases like they are strongly soluble in water, absorbs light in UV region at 260 nanometer, capable of forming hydrogen bonds. So we all know that they form the double bond or triple bond, and pyrimidines are also soluble at body temperature or body pH, also observe UV light at 260 nanometer, and they are also, the pyrimidines are also capable of forming the hydrogen bond. So these are the pyrimidine bases. So are you able to get it? Everything is clear till now. It is very basic thing. I hope all of you will get it definitely. I will share this PPT with all of you so that you will be able to read it again and again when possible. These all are the metabolism, the metabolic pathways which we have to discuss later. So I will not present, or I will not let you know right here, but we will definitely discuss it later on within the coming lecture series. We will discuss these all things. Then these are the functions of nucleotides. You can read it by your own self also. So is it clear till now? Is it clear to all of you? Please let me know. Is it clear to all of you? Please let me know till now. Because you have to remember the bonding, how DNA is formed, DNA's helical structure, which type of bonding is there, where there is a double bond, where there is a triple bond. Everything is clear till now. Please let me know. Like this is again Watson and Crick base pairing, that guanine and cytosine, they will form the triple bond, adenine and thymine, they will form the double bond. Now this is the replication which we have to discuss. We will try to discuss it today, or we will do it tomorrow also. So up to now, is it clear to all of you? It's a very simple thing. You all know about the transcription, translation, but we have to discuss it in very detail also. So we have to discuss these all things in detail also. But up to now, is it clear to all of you? Please let me know. I have one video of this also. I want to share with all of you. Just let me know whether it gets played or not. It will take some time. Any question, anything you want to know regarding the review paper, please let me know. About the review paper, if you have any doubt, please let me know. Have you selected a topic, or I have to distribute those topics to you? Please let me know. Have you selected the topic or not? The review paper topic, have you selected or not? Please let me know. By 10th of December, you have to submit me the abstract. Please let me know. Or I have to share with all of you. Please tell me whether you will do it by your own self, or I have to share. It's not opening right now. Have you selected the topics of review paper?
Do you recognize this molecule?
Please let me know.
This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly.
I think the volume is not audible to any of you. Okay, the volume is not audible. I will share this video with all of you in the group because we are not able to get the volume properly. So that is why. Okay, you all need that. I will send you some topics for review paper. You all want that I will send you some topics for review paper, or you will do it by your own self. Okay, I will send some topics related to biochemistry in your group. You have to select any of the topic and you have to write the review paper on it. Is it clear to all of you? Please let me know. All of you, please let me know. All of you. No, 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 it's not compulsory for any of you to select the biochemistry topics. No, it's not compulsory, but I will let you know about 10 topics or 10 topics related to biochemistry in your group, or even you can select your own topic and you have to send the topic, the abstract on my email ID so that I will check it and I will let you know about it. Okay, good, good. If you have selected a topic, then please, 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 after this session, you have to be in touch with me and let me know about the topic on which you are working. Getting my point? So after this lecture series, you have to be in touch with me and you have to let me know on which topic you are working. Yes, you have to write the review paper, Sonal. I will let you know it by tomorrow. Generally, you have to go on Google Scholar. Let me show you. You have to go on Google Scholar. Okay, Google Scholar. Suppose I want to do a review paper on COVID. We will write COVID-19 COVID-19 pandemic in India review. So I just write this thing. Now I will enter search. So I am able to get so many. I am able to get 59,500 papers within 1 minute. So you can go next, next. So you have to open those papers. So I am opening this. That is review of forecasting models in COVID-19 pandemic in India during lockdown. So I will have to open this review paper and I have to read this paper. These are also the students like you, like Abhinav, Ravi. They all are the students. They are from New Delhi, India. So you have to read their abstract, that how they are making the abstract and on which thing they are working. Like they said, COVID-19 pandemic, also known as coronavirus pandemic, is currently wreaking havoc in more than 200 countries. So you have to select the paper like a review of modern technologies for tackling COVID-19. You can even go for 2016 also. So there are some papers which are there. You have to open, you have to download all these papers. Mental health problem faced by workers during COVID-19. This is also a good paper, that how the workers, they are facing the problem during the COVID-19 period. Even you can write anything like suppose breast cancer in India. Anything you can write. So you will able to get so many papers related to breast cancer. Breast cancer survival studies in India, a review. So someone like you, some students are there. They worked on the cancer patients and they write the paper on breast cancer survival rate. So you have to download these types of review papers. This is abstract. You have to read all these papers and then you have to write your own paper, or you can say molecular markers. Molecular markers review. So you have to importance of molecular markers in livestock, application of molecular markers in plant genome. So you can download those papers which are your point of interest, and you have to compile these paper in your own paper. Getting my point? So now you are asking so many questions, you are not ready to listen to me first. How the teacher will ask you to copy paste any other's work? Is it possible? Is it possible for me that I will tell you how to copy paste? It is not like that, dear. You have not to copy paste. When you are taking anyone's line, you have to add the reference just after that particular line. So instead of asking questions and questions, first listen what the teacher is saying. Getting my point? First you write how do we write a review paper. Now I just tell you how to get the knowledge of writing your own review paper. For writing a review paper you have to read at least 19 to 20 papers. So read those papers and then you have to make abstract of your own. It is a review paper, it is not a research paper. In review paper, you have to add the work, the contributions which is done by the other people, not by your own self. Getting my point? So I think you all will get it right now. If you want, I will share the topics with all of you. Otherwise, you can do it by your own self also. You have to type it on Google Scholar. You will get so many research papers. You have to read those papers again and again and again and again. Then you have to compile, you have to club those work together in your own review paper. I hope it will be clear to all of you. We will meet tomorrow. Till then, take care of yourself, and till then, search any topic and you can WhatsApp me at any time so that I will be able to help you. So I am there only for helping you all, but I just want that you select the topic by your own self, by your point of interest, how you want to do, and then we will start. No issues, I will ask you, I will help you, but you have to select the topic first. So I think that's all for today. We will meet tomorrow. Till then, take care of yourself and be happy. So be happy all of you. Please, it's my request to all of you. Please don't panic. Okay, this is a 20-day journey. This is a 20-day lecture series, and we will do it slowly and slowly and slowly. So please don't panic or something like that. I will help all of you. Please, we will do it very slowly and slowly. Okay, so no issues, no worries. We will do very slowly. I am there along with you, not only for these 20 days. Rather, we will be together up to your paper gets published. At that time we will be together. So please don't worry, don't panic, please, any of you. Okay, getting my point? So till then, take care of yourself. We will meet soon. Bye bye and enjoy your day. And just focus on the paper, the topic on which you want to write the review paper. You have to tell me by 8 p.m. today. I will be there in the Telegram, and then we will chat with each other. Okay. So please take care of yourself. Please don't get panic. We will do it very slowly and slowly. And I will be there along with you up to your paper gets published. So don't worry. Please be relaxed. Any another thing you want to know, please let me know. And if any another doubt is there, please let me know. If you have any another doubt, any of you, please let me know. But please don't panic. Any another question or query if you have, please let me know. And yes, you have to draw the diagrams of purines, pyrimidines, nucleotides, nucleosides. I will check it in the Telegram also. Yes, Narita, it will be same. Do you have any problem regarding the timing? or do you have the class at 3 p.m.? Please tell me. Have you any class right now, Narita? If you have any class, please let me know. Please, if you have any problem, please share with me. No issues, no problem. No, if you have any class, then please share. Please share. Yes, Tejasni, you all are free. You can select from anywhere, anywhere. Ayushi, I will discuss it with the authorities. If they will allow me, then we will take it on Google Meet. Okay. So no worries. I will talk with the Edufabrica team and then I will let you know. Getting my point? Any another thing, any another question, please let me know, please. Radha has classes up to 4. Radha, just try to manage because even on weekends we will shift the timings. On Saturday, Sunday, I will take it on different timings. So I just request you, is it possible to just wind up your classes by 3 p.m. If it is possible, then please let me know. I hope you will understand, Radha, and you will definitely cooperate. In the meantime, think of uh, yes, Ayushi, I understand. Yes, Narita, we will do the discussions, but not today. On Saturday and Sundays, I have ample of time along with me. Then we will do everything. Okay. Regarding, I will introduce myself and I will also take the introduction of yourself also. So, pardon me today, we will discuss it on weekends. Now, please select any topic and let me know. Okay. So till then, take care of yourself. Radha, please try to be there by 3 or 3:20 p.m. if it is possible. And also let me know if you are comfortable right now. So if you are able to listen to me, then please reply. Radha, do you have any type of exams in your class? Please let me know. You have any types of exams right now in your class? Like practical or something like that, please let me know. No, you discuss it with me also. I am there for helping you all. I am just asking you whether you have your exams or something which is there like from 3 to 4 p.m. Do you have any exams or something like that? Please share with me if possible. Okay, so you have your practicals. Okay. And you are free on Saturday and Sundays, or you have the busy schedule on weekends also? Please let me know. You have 5 days working in your institute, or it is 6 days working? Okay, you are free on weekends. Okay. No issues. We will try to do our best. And we will try to do our class by 3:30 because till then, all of you, I request all of you to just focus on the review paper and let me know on which topic you want to work. Okay. So I think that's all from my side. We will meet tomorrow. Okay. Till then, take care of yourself. Bye bye.